Hello, my name is Tay Netoff, and I'm an assistant professor of biomedical engineering at the University of Minnesota. Today I'm presenting a paper titled Chaotic Desynchronization is the Therapeutic Mechanism of Deep Brain Stimulation. My co-authors are Charlie Wilson and Bryce Beverland. This paper was published in 2011 in Frontiers in Systems Neuroscience as a hypothesis and theory article. Deep brain stimulation is where they take an electrode and plant it in the brain to stimulate electrically different regions for alleviating symptoms of Parkinson's disease or depression or epilepsy. The mechanism by which deep brain stimulation works is not well understood. So there's some experimental evidence out there showing how neurons respond to deep brain stimulation and some modeling work that's come out, which is really interesting. This paper shows some alternative hypotheses. Our research was originally inspired by this paper in 2003 by Hashimoto et al., where they took electrodes implanted in monkeys that were made Parkinsonian, stimulated, and measured single unit to identify when they fired with relationship to the stimulus. And you can see in panel A in the left, when the stimulator is off, fire uniformly with respect to the stimulus times. But when the stimulus is on, you can see the neurons cluster at certain phases. Why they cluster at those phases is not exactly clear. One hypothesis is that the stimulus elicits a volley of EPSPs followed by excitation of inhibitory neurons, so you get a volley of IPSPs, and then you get a release from those IPSPs to get another volley of action potentials and EPSPs. But we're going to present another hypothesis that shows this kind of distribution explained by the dynamics of the neurons themselves of the targets to the stimulus. Alan Dorval in 2010 did a great experiment where they took patients that were having their batteries replaced for deep brain stimulation used for Parkinson's, and they had them do a finger tap test where they were tapping periodically, and when the symptoms are bad, it's difficult for them to tap, so the tap duration gets extended and the interval variability is fairly high. When they turn on the stimulus, then the tap durations go down and become more regular. And what Alan decided to do was test to see if by making the stimulus period a little more irregular, whether that improved or decreased the efficacy of the stimulus. And he found that the increased variability of the stimulus was less effective, as you can see on the 130 plus or minus 60% was less effective while maintaining the same average period. In 2004, Jonathan Rubin and David Terman made a nice model of the basal ganglia during deep brain stimulation. The basis of their model was that neurons normally don't fire synchronously, as you can see in the left-hand side of this figure with A and B. These are two neurons modeled in the subthalamic nucleus, and they are not synchronously firing. But in the Parkinsonian state, they start firing in clusters, and those clusters then alternate between the two neurons, and you get oscillations. And those oscillations tend to block activity in the thalamus from letting motor signals pass through. Deep brain stimulation regularizes the firing patterns of the neurons in the subthalamic nucleus, which then allows the activity passing through the thalamus to pass uninhibited. Our paper takes a slightly different approach, not assuming that the neurons are stimulated to fire by the deep brain stimulation, but instead the neurons are already firing, and the deep brain stimulation just modulates their firing time. And at certain stimulus frequencies and amplitudes will show that the stimulus can actually synchronize populations or desynchronize populations. We will use phase response curve theory to demonstrate how and when the neuronal spike times of neurons in response to deep brain stimulus becomes chaotic. And a stimulus input applied here at some phase phi causes a change in the neuron's spike time from its natural period. If we stimulated a different phase, you can see that actually delays the next spike. So depending on the stimulus phase, you get a different response from the neuron. And by stimulating at a bunch of different phases and measuring this change in the spike time as a function of the stimulus. In the first figure of the paper, we show how you can predict the phase of the stimulus on the next cycle from the phase of the stimulus on this cycle. And so the stimulus comes in and advances the spike, so that changes the stimulus phase on the next cycle. From that figure, you can see the stimulus phase, TRI+1 on the next cycle divided by TC, which is the period of the cell, is equal to the phase on the previous cycle, TRI, divided by the period, plus MS, which is the stimulus amplitude of the PRC. In the argument to the PRC is the period 1 minus TRI over TC, which is the phase of the stimulus. And then we add the period, which is 1, minus the ratio of the stimulus period to the cell's period, which tells us how much change you see in the phase just by differences in the stimulus period. If we set the PRC amplitude to 0, and the stimulus period and the neuron's period are the same, we'll see that the phase of the stimulus on the next cycle is equal to the phase of the stimulus on the previous cycle. In figure 2, we demonstrate here are two neurons that are uncoupled and they're just firing periodically. And then when we turn on a stimulus and update the spike times according to the phase response curve, and the phase response curve is given here in C. So at early in the phase, there's very little advance of the spike times, whereas about 2/3 of the way through the phase, we get about a 10% phase advance. So when we turn on the stimulus, you can see it changes the phases of the neurons, and the relative phase, which is plotted here in B, you can see is constant, and then the moment the stimulus is turned on, the relative phase starts changing. In figure D, we can graphically demonstrate how the phase of the next cycle can be determined from the phase of the last cycle. So phi here is the phase of the stimulus on this cycle, and we can say, for example, the phase starts here at 0.3, and then we go up to the blue line and map that over, and it tells us what the phase will be of the next cycle. And you can see where the blue line crosses the red line being the line of identity, meaning that the phase on this cycle is exactly equal to the last cycle, is those crossing lines are the fixed points. And this one is an unstable fixed point, and the one at 0.8 is a stable fixed point. By changing the stimulus frequency, we actually move this blue line up and down. And at some point, these lines no longer connect, and then when we map the phase on the next cycle just from the last cycle, we can do what's called a cobweb. So you go up to the blue line and then over to the line of identity, it wraps around, and then back down to the blue line to determine what the stimulus will be on each subsequent cycle. And you can see when in D, the stimulus phase locks in at 0.8 and stays at that, so the neurons are entrained to the stimulus, whereas in E, when there is no connection, the neurons are constantly sort of moving around with respect to the stimulus. F is an example of the actual phase difference between the stimulus and the neuron over many, many cycles, and you can see when there's no connection between the blue and red line, there's very complex behaviors. So down here in G, we've actually sort of measured the complexity of the neuron's response using a measure called the Lyapunov exponent, which we'll explain a little bit more in detail on the next slide. And entrainment we plot as white here. When it's very weakly entrained, we plot it at blue. And when we get this behavior that you see in F, which is chaotic or could be chaotic, we plot it in orange. In G, we're showing the response of the neuron as a function of stimulus frequency and stimulus intensity, showing large ranges of entrainment and small ranges of chaotic behavior to excitatory inputs. And on the right-hand side, we show the response of the neuron to inhibitory inputs. And at very low stimulus amplitudes, you see very little interaction between the stimulus. Now, one question we had was, this very fine structures that you see here looks fractal, but neurons are noisy, and this was a deterministic model. And in noisy neurons, does this fractal behavior go away and we lose the positive Lyapunov exponent areas, or do they stay positive? In figure 3, we're measuring phase response curves that are noisy. So here's the phase response curve with the standard deviation around it. The standard deviation was estimated based upon the phase response curve according to the paper by Ermentrout et al. In 2011, which I'm a co-author on. And in B, we make a stochastic map, which shows the phase response curve just like we saw in the previous figure, but it's a probabilistic distribution. And from these maps, we can calculate what the steady state distribution of the phases with respect to the stimulus that we would expect.